divided by one gene and two alleles, a dominant allele and a recessive allele, in the tasks that follow. Three more simplifications are also made. Complete penetration. When an individual in the pedigree possesses at least one dominant allele of a dominant trait or two recessive alleles of a recessive trait, the person will be affected to display the phenotype linked to a trait. Rare in population. The relevant feature is uncommon in the general population in each of the problems. For the purposes of these issues, let's assume that third and second generation marriages into the pedigree don't result in carriers. Since either or both of the people at the top of the pedigree could be carriers, this does not apply to the founding parents. Not Y-linked. These issues may have autosomal or X-linked causal genes. Y-linked genes are not involved. 5 major hints. When it comes to pedigrees, there are 5 factors to keep in mind. No allele of a dominant trait can be present in an unaffected person. Because each gene are working and making the right gene product, they will not develop signs and symptoms of the genetic condition caused by this gene not working properly. Black small square. When this person has a child, they will only pass on a working gene copy, R, to each of their children. White circle. Genetic carrier is the one who does not have the condition unaffected. Black small square. This person has a gene pair, RR. This means that one copy of the gene is working and making the right gene product, however the other copy, 1, is not working. They will generally not develop signs of the genetic condition because, for the correct expression of this gene, you only need one working copy. This person is a genetic carrier of the genetic condition caused by this abnormal gene as they carry the defective gene and can pass it on to the next generation. Black small square. When this person has a child, there is a 50% chance they will pass on a working R gene copy to each of their children and a 50% chance they will pass on the gene variant, which is not working R. White circle. Affected or more likely to develop an autosomal recessive condition. Black small square. This person has a gene pair, RR. This means that both copies of the gene are not working and they are unable to make the gene product. They will develop signs of the genetic condition caused by this defective gene. Patterns that indicate a dominant trait. If every affected child of non-founding parents has an affected parent, the disease must be dominant. The unaffected woman, who will soon become pregnant, does not have the disease. Therefore, the affected child can only acquire an allele from the affected father. The trait is dominant because no kid may be impacted by a single autosomal recessive gene or X-linked recessive allele. The disease must be autosomal dominant when an affected son of non-founding parents has an affected father. The disease cannot be X-linked dominant since a parent cannot pass X-linked alleles to his son. We cannot tell if the dominant disease is X-linked or autosomal when an affected daughter of non-founding parents has an affected father. Both an X-linked dominant allele and an autosomal dominant allele can be passed on to the affected father's daughter. Autosomal recessive inheritance. Autosomal recessive inheritance. The non-working copy of a gene is represented by R and the working gene copy by R. There are 3 possible ways this may show up in a person, both in case of a male or female. Figure 2. White circle. Non-carrier is one who does not have the condition unaffected. Black small square. This person has a gene pair, RR. This means that both copies of the black small square of this person has a child, they will only pass on the gene variant which is not working R. Figure 2. Autosomal inheritance patterns. Examples. Albinism, a complete depigmentation of the skin due to deficiency of tyrosinase. Phenylketonuria, caused due to lack of phenylalanine hydroxylase and manifested in the form of mental retardation, fair skin, and epilepsy. Sickle cell anemia, the beta chain of hemoglobin contains valine in the 6th position instead of glutamic acid. Autosomal dominant gene inheritance. Here, an autosomal dominant gene variant is represented by D, and the working gene copy by D. There are 3 possible ways in which this may show up in a person regardless of whether they are male or female. Figure 3 and figure 4. Figure 3. Autosomal dominant variants, A and C affected and DD normal in this case. Figure 4. Autosomal dominant inheritance patterns. Examples. Neurofibromatosis type 1. Achondroplasia. Huntington's disease, neural degeneration leading to convulsions and premature death. Late onset disease. Inherited likelihood to develop breast, ovarian, and bowel cancers. Familial hypercholesterolemia. Brachydactyly, shortening of fingers. Polydactyly, occurrence of extra fingernail patella syndrome, dystrophy of nails, absence of patellae, and nephropathy. Sex-linked recessive inheritance. Here, a non-working X-linked recessive gene copy with a variant is represented by R and the working gene copy by R. There are several possibilities for a person depending on if they are male or female. The variants and inheritance patterns are illustrated in the figure 5 and figure 6. Figure 5. Sex-linked recessive variants. Figure 6. Sex-linked inheritance patterns. Example. Hemophilia, caused due to functionally defective antihemophilic globulin. Duchenne muscular dystrophy. Color blindness, inability to distinguish different colors, red protein type and green Duden type. Testicular feminization syndrome, 44 plus XY male, phenotypically female. Fragile X syndrome. Sex-linked dominant inheritance. Where an X-linked dominant gene variant is shown as D, and the working gene copy by D. There are several possibilities a person could have, and depending on whether they are male or female, the effect of the genetic makeup will differ. Figure 7 and figure 8. Figure 7. Variants of sex-linked dominant inheritance. Figure 8. Sex-linked dominant inheritance patterns. Example. Hypophosphatemia, a type of vitamin D-resistant rickets. XG blood group. White circle histiocytosis X is characterized by the presence of cytoplasmic rod structure called Langerhans cell granules or X granules XG. White circle hypogammaglobulinemia and measles. Pneumonitis. White circle faulty tooth enamel and dental discoloration. Y-linked polydric inheritance. Only males are affected. An affected male must transmit the disorder Y-linked traits to all of his sons but none of his daughters. It is also known as polydric inheritance or transmission. Examples. Hairy pinnae. Bilateral radio ulnar synostosis, radial layer fused. Mitochondrial inheritance. There are peculiar patterns in the diseases brought on by mitochondrial DNA mutations. White circle. Both sexes are affected, and the disorder is passed down through the female to her progeny. If a guy has a trait and his spouse does not, their progeny will not also have the characteristic. White circle illustrated in figure 9. Figure 9. Mitochondrial inheritance. Log score for linkage testing. The logarithm of the odds log is a log score and is symbolized as C. The most effective statistical technique for assessing linkage analysis in a pedigree study was demonstrated by Newton Morton in 1955. Log score is a statistical estimate of whether 2 genes or gene in a disease gene are likely to be located close to each other on a chromosome and are therefore likely to be inherited together as a package. Log score. Criteria. A large data sample is needed to determine the log score. White circle. The bigger the sample, the better. White circle. Bigger sample yields more meaningful information statistically. The analysis would be less ambiguous if the families are with a large number of children. For pedigree analysis, at least 3 generations are to be taken. The method simply calculates 2 different probabilities for obtaining a set of results in a family. The first probability is by assuming independent assortment and the second is by assuming a specific degree of linkage. Then the ratio odds of the 2 probabilities is calculated and the logarithm of this number gives the log value. Human genetics calculate a log score to confirm or reject co-segregation. Log score. Calculation. It is based on the ratio of the probability of observing co-segregation if the genes are linked and the probability of observing co-segregation if the genes are not linked. Probability of observing co-segregation if the genes are linked. Log score Z. Probability of observing co-segregation if the genes are not linked. Equals OR 1 and RXR. Computational limits regarding the analysis of types of pedigree. It is necessary to specify a precise genetic model and mode of inheritance, the penetrance of each genotype, and gene frequencies. Karyotypes. The complete set of chromosomes of an organism is called karyotype and is usually presented as a picture of metaphase chromosomes lined up in descending order of their size. Each functional chromosome has a centromere, where spindle fibers attach, and 2 telomeres that stabilize the chromosome. Chromosomes are classified into 4 types: white circle metacentric, centromere in the middle. Equal arms. White circle submetacentric, centromeres displaced towards one end, short and long arm. White circle acrocentric, centromeres near one end, a long arm and a knob or satellite. White circle telecentric, centromeres that are very near the end of the chromosome. The short arm is denoted by P and the long arm is denoted by Q. Karyotype. Preparation. Cells, stages, staining, preservation, and analysis. Karyotypes are prepared from actively dividing cells, such as WBCs, bone marrow cells, or cells from mersomatic tissues of plants. After treatment with a chemical, e.g., colchicine, that prevents them from entering anaphase, the cells are chemically preserved, spread on a microscope slide, stained, and photographed. The photograph is then enlarged, and the individual chromosomes are cut out and arranged in a karyotype. NR equals non-recombinant progeny. R equals recombinant progeny. Theta equals recombinant fraction, fraction of birth in which recombination has happened between the studied genetic marker and the putative gene associated with RNR plus R. Reason for using 0.5 in the denominator: alleles that are completely unlinked have a 50% chance of recombination due to independent assortment. Log score, significance. Because there might be a lot of crossover during meiosis, the number is typically stated logarithmically when genes are on different chromosomes. For example, the process of cell division by which sperm and egg cells are formed. If genes are on the same chromosome but far apart, they are also inherited independently of each other. Although, when log scores are in close proximity on the same chromosome, they tend to be inherited together. White circle if the log score is high plus bay log traits. Are closely linked and inherited together. White circle if the log score is low bay log traits. Are unlinked and not inherited together. White circle if the log score is 3 or higher high. Probability of genetic linkage. Lower number, less probability of genetic linkage. White circle odd equals plus 3 and 1000 over 1 odd of the linkage. Being observed did not occur by chance. White circle odd is greater than or equal to plus 3 significant evidence of linkage. White circle odd is less than or equal to minus 2 significant evidence of non-linkage. White circle is less than or equal to odd is greater than or equal to 3.0 insufficient data. Log score, problems. Problems with locus heterogeneity, vulnerability to errors. Human karyotype, figure 10. Figure 10, human karyotype. Karyotype, preparation and staining banding patterns described later in the chapter. G banding. Q banding. C banding, R banding. Karyotype, uses of chromosome banding applications. G and R banding are the most commonly used techniques for chromosome identification, karyotyping, and for identifying abnormalities of chromosome number, translocations of material from one chromosome to another, deletions, and inversions or amplifications of chromosome segments. This has had an invaluable impact on human genetics and medicine, and the power of this approach has been augmented by combining cytogenetics with fluorescence in situ hybridization fish. The detection of chromosome deletions associated with disorders, very often contiguous gene syndromes, provided some of the first disease gene localizations in humans. Translocations have been important in pinpointing the location of disease-associated genes, and the characteristic translocations associated with some leukemia are important not only for understanding the molecular basis of these cancers but also for their diagnosis and prognosis. White circle, one of the best examples of this is the translocation between human chromosomes 9 and 22, 9, 22q34, q11, or the Philadelphia chromosome diagnostic of chronic myelogenous leukemia, CML. Comparisons of chromosome banding patterns can confirm evolutionary relationships between species and also reveal changes in karyotype that may have been important in speciation. The banding patterns of human, gorilla, and chimpanzee chromosomes are almost identical, though human chromosome 2 is the result of a fusion between two grade 8 chromosomes. G banding, figure 11. G banding, or Gien's banding is a technique used in cytogenetics to produce a visible karyotype by staining condensed. G banding, or Gien's banding is a technique used in cytogenetics to produce a visible karyotype by staining condensed chromosomes. It is useful for identifying genetic diseases through the photographic representation of the entire chromosome complement. The metaphase chromosomes are treated with trypsin, partial digestion of chromosome, and stained with Gien's stain. Heterochromatic regions, AT-rich, in DNA are relatively gene poor and stain more darkly in G banding. In contrast, less condensed chromatin, euchromatin, GC-rich, are more transcriptionally active and incorporate less Gien's stain, and these regions appear as light bands in G banding. The pattern of bands is numbered on each arm of the chromosome from the centromere to the telomere. This numbering system allows any band on the chromosome to be identified and described precisely. The reverse of G bands is obtained in R banding. These bands were same in appearance on the homologous chromosomes, thus identification became easier and more accurate. The less condensed the chromosomes are, the more bands appear when G banding. This means that the different chromosomes are more distinct in prophase than they are in metaphase. There are also extensive similarities between human chromosome bands and those of lower primates. What it detects: microscopic genomic abnormalities and GT 5-10 megabits, white circle inversions, white circle duplications, deletions, white circle balanced and unbalanced translocations, white circle aneuploidies, and GT 10% mosaicism. For example, cultures where in GT 1 of 10 cells are trisomy 12, what it doesn't detect: submicroscopic genomic abnormalities and LT 5 megabits. When to use: as a baseline genomic screen, white circle at derivation of cell lines. White circle at the start of experimental protocols, white circle to assess and monitor genomic stability. E.g., every 5 to 10 passages of cell culture, white circle at the conclusion of experiments prior to publication. For cell line banking, when publication, quality karyotypes are needed. Banding type staining method: C banding constitutive. Heterochromatin, G banding, Gien's sustain, Q banding, quinaprine. R banding, reverse Gien's sustaining. T banding, telomeric. Table 1 banding types and staining methods for karyotype studies. Figure 11, G banding. Q banding, fluorochrome banding, figure 12. Fluorochrome specific for particular DNA. Bases are historically important but not used. Now in routine cytogenetics is the banded. Preparations are not permanent. The most useful counterstains for fish are DAPI and chromomycin E3. Staining, chromosome preparation is immersed in a dilute solution of fluorochrome for a short time mounted to retard fading. Q banding, use of quinaprine banding similar to G banding. Mipramycin similar to chromomycin E3 gives a pattern similar to R banding. Figure 12, Q banding. Early condensation. Packaging chromosomes. Little recombination. Nuclease insensitive. Low concentration of gene. Low histone acetylation. Chromosome preparation fixed with alcohol-acetic acid defernates DNA are treated successfully with DIL. Acid defernates DNA, alkyl O2 hydrolysis of defernated DNA, warm saline extraction of hydrolyzed DNA, and stained with Gien's selective stain for constitutive heterochromatin. R banding, reverse banding. R banding is a cytogenetic technique that produces the reverse of the G band stain on chromosomes. R banding is obtained by incubating the slides in hot phosphate buffer, followed by treatment of Gien's dye. The resulting chromosome patterns show darkly stained R bands, the complement to G bands. Darkly colored R bands are guanine cytosine rich, and adenine thymine rich regions are more readily denatured by heat. The technique is useful for analyzing genetic deletions or chromosomal translocations that involve the telomeres of chromosomes. Characteristics of euchromatin bands in mammalian chromosomes. G bands. Positive GQ bands. Negative R bands. At rich DNA, late replicating DNA. Quantitative genetics 8. Genotypes are possible, each producing a slightly different phenotype. They are influenced by the environment. White circle quantitative characteristics often arise when environmental factors affect the phenotype because environmental differences result in a single genotype producing a range of phenotypes. Relation between genotypes and phenotypes, table 1. Plant. Genotypes, doses, height, cm. AABBCC 010. AABBCC, AABBCC, AABBCC. 111. AABBCC, AABBCC, AABBCC. 212. AABBCC, AABBCC, AABBCC. A branch of genetics which deals with the study of the inheritance and genetic analysis of quantitative or polygenic characteristics that are determined by multiple genes is called quantitative genetics. The analysis of quantitative genetics is based on means, variances, and covariances. Comparison between qualitative and quantitative traits. Qualitative traits. It is influenced by a single gene. The phenotype of this trait falls into distinct categories: presence or absence of disease. Discontinuous variation and distinct phenotypic classes are present. Typically follows a simple pattern of inheritance. Trait expression is typically unaffected by environmental factors. Quantitative traits. It is influenced by multiple genes. The phenotype of this trait is expressed on a continuum. Characters are expressed in terms of degree. Also described as a continuous variation or phenotypic measurement from a spectrum. Does not follow a simple pattern of inheritance. Trait expression may be affected by environmental factors. Quantitative traits. Two phenomena characterizing quantitative traits. They are polygenic white circle. They are influenced by genes of many loci. If many loci take part, many quantitative traits. Inference figure 1. Figure 1. Quantitative traits are demonstrated by a bell-shaped curve, and it is not possible to know whether an individual has a certain phenotype due to one genotype or the other continuous and discontinuous traits. Types of quantitative characteristics. This trait demonstrates true continuous variation, i.e., from one phenotypic extreme to the other, from minimum to maximum, with the non-existence of clear-cut boundaries. There is a continuous gradation from one phenotype to the next. They are influenced by environmental factors. Examples: weight of an organism, blood pressure of human, and IQ. Figure 2. Figure 2. Continuous traits. The IQ scoring method uses an average score of 100, with individual scores distributed according to a Gaussian distribution, percent on y-axis. Meristic trait. This type of trait does not demonstrate continuous variation. The phenotypes are placed into discrete categories due to the intrinsic nature of the character. The value of the phenotype of such a trait is determined by counting. This is why they are known as countable characters. Examples: number of skin ridges forming fingerprints and the number of bristles in the abdomen of a fly. Threshold dichotomous trait. These quantitative traits exhibit only two or a few phenotypic classes. Their inheritance is determined by multiple genes and environmental factors. Examples: diabetes and schizophrenia. Quasi-continuous trait. The quasi-continuous trait feature represents continuous variables over one range of it, and then represents categorical data over the rest of it. An example is shown in the figure 3. Figure 3. Quasi-continuous trait. Advantages and limitations of quantitative genetics. Advantage. It provides a solution to the analysis of polygenic characters which is not possible through Mendelian genetics. Limitations. Polygenic characteristics are sensitive to environmental variation. Therefore, quantitative estimates are valid for a particular set of environmental conditions only. White circle occurs when parents are intermediate to the extreme values of the segregating population. Parents: AABB x AABB gametes, A flat F1 AABB. F2 AABB x AABB. F2 AABB and AABB transgressive. Segregants. Examples of polygenic inheritance. Kernel color in wheat, skin color in man. Kernel color in wheat. Whole grain or seed of a cereal plant, corn in wheat, is called a kernel. Kernel color in wheat is a quantitative trait and is controlled by two different genes. Nielsen L. 1908 studied the inheritance of kernel color in wheat. These estimates may change with the change in environment. Polygenic inheritance. Polygenic inheritance has been reported for various characters in plants and animals. It includes kernel color in wheat and skin color in man. They show a normal or bell-shaped curve. Transgressive segregation and transgressive segregant. The phenomenon of appearance of F2 individuals with a lower or higher intensity of character than those present in both the parents involved in the cross is called transgressive segregation. Segregants that fall outside the range of both the parents for a character are known as transgressive segregants. Features white circle transgressive segregants are only possible from the crosses between two parents having mean value for a quantitative character. White circle transgressive segregation results due to the fixation of dominant and recessive genes in separate individuals. Genotype genotypic. Each of the contributing alleles, RC, adds some red to the phenotype of kernel color so that genotypes of the white kernel contain neither of these alleles. The seed color was purple when four effective or contributing alleles were present. Similarly, where three contributing alleles were present, the kernel color was dark red. In the presence of two effective alleles, the color of kernel was red, and with one contributing allele, the kernel color was light red. White kernel color was developed in the absence of all the non-effective alleles. The phenotypic ratio of the F2 is 1, 4, 6, 4, 1. This ratio is a product of binomial expansion. Skin pigmentation in humans. Skin pigmentation in humans varies from light to dark. It is controlled by three genes. C.B. Davenport 1913 observed that two pairs of genes, A and BB, cause the difference in skin pigmentation between Negroes and Caucasians. These genes were found to affect the character in an additive fashion. Thus, a true Negro has four dominant genes, AABB, and Caucasians have four recessive genes, AABB. Requirement: white circle Negroes, AABB. White circle Caucasians, AABB. Procedures: white circle cross between Negroes and Caucasians gives birth to a child with an intermediate skin color termed mulattoes. White circle mating of two such mulattoes will produce a wide variety of skin colors in offspring: dark Negro to Caucasian. Parent Negro black, AABB, X Caucasian, AABB, gamete ABAB. F1 AABB, mulatto white pointing arrow medium, intermediate skin color, F2 equals F1 AABB, X F1 AABB. Gametes, AB, 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 and AB in both the parents. Genotype genotypic. Heritability is a measure of resemblance between relatives and is estimated by evaluating the degree of resemblance, i.e., observed phenotypic differences variance of a trait are controlled by inheritance genetic. It is important to know how much of the variance in a quantitative trait is due to genetic differences and how much is due to environmental differences. That proportion of the total phenotypic variation that is due to genetic differences is known as heritability. Phenotypic variance. To determine how much phenotypic differences in a population are due to genetic and environmental factors, we must first have some quantitative measure of the phenotype under consideration. Consider a population of wild plants that differ in size. We could collect a representative sample of plants from the population, weigh each plant in the sample, and calculate the mean and variance of plant weight. This phenotypic variance is represented by BP. Components of phenotypic variance. 1. Phenotypic variance. Phenotypic differences among individual members of a group can be attributed to several factors. 2. Some of the differences in phenotype may be due to differences in genotypes among individual members of the population termed genetic variance VG. 3. Some of the differences in phenotype may be due to environmental differences. These differences are termed environmental variance A. Determining gene number for a polygenic characteristic. When two individuals homozygous for different alleles at a single locus are crossed A1A1XA2A2 and the resulting F1 heir interbred. If the original parents are homozygous for different alleles at two loci, as are those in Nielsen-Ells crosses, then 1/4 times 1/4 equals 1/16 of the F2 should resemble one of the original homozygous parents. Generally, 1/4 n will be the number of individuals in the F2 progeny that should resemble each of the original homozygous parents, where n equals the number of loci with a segregating pair of alleles that affects the characteristic. This equation provides us with a possible means of determining the number of loci influencing a quantitative characteristic. Illustration. Let us assume that we cross two different homozygous varieties of pea plants that differ in height by 16 centimeters, interbreed the F1 and find that approximately 1/256 of the F2 are similar to one of the original homozygous parental varieties. This outcome would suggest that four loci with segregating pairs of alleles, 1/256 equals 1/44, are responsible for the height difference between the two varieties. Because the two homozygous strains differ in height by 16 centimeters and there are 4 loci each with 2 alleles, 8 alleles in all, each of the alleles contributes 16 centimeters, 8 equals 2 centimeters in height. This method for determining the number of loci affecting phenotypic differences requires the use of homozygous strains, which may be difficult to obtain in some organisms. It also assumes that all the genes influencing the characteristic have equal effects, that their effects are additive, and that the loci are unlinked. 9. Dominance genetic variance BDA. This is observed when some genes have a dominance component. B. In this case, the alleles at the locus are not additive. Rather, the effect of an allele depends on the identity of the other allele at that locus. C. Here, we cannot simply add the effects of the alleles together. D. Instead, we must add a component BD to the genetic variance to account for the way that the alleles interact. E. Also, the genes at different loci may interact in the same way that alleles at the same locus interact. F. When this genic interaction occurs, the effects of genes are not additive, and we must include a third component called genic interaction variance BI to the genetic variance. IPG equals BA plus BD plus BI. 10. Additive genetic variance BAA. It comprises the additive effects of genes on the phenotype, which can be summed to determine the overall effect on the phenotype. B. For example, suppose that, in a plant, allele A1 contributes 2 grams in weight and allele A2 contributes 4 grams. C. Example, kernel color in wheat. Summary equation. 1. 1. We can now integrate these components into one equation to represent all the potential contributions to the phenotypic variance. APP equals BA plus BD plus BI plus BD plus BGE. 2. This equation provides us with a model that describes the potential causes of differences that we observe among individual phenotypes. 3. It's important to note that this model deals strictly with the observable differences variance in phenotypes among individual members of the population. It. 4. Environmental variance includes differences that can be attributed to specific environmental factors, such as the amount of light or water that the plant receives. 5. It also includes random differences in development that cannot be attributed to any specific factor. 6. Any variation in phenotype that is not inherited is, by definition, a part of the environmental variance. 7. Genetic environmental interaction variance BGE arises when the effect of a gene depends on the specific environment in which it is found. A. For e.g., in a dry environment, genotype A produces a plant that averages 12 grams in weight and genotype A produces a smaller plant that averages 10 grams. B. In a wet environment, genotype A produces a larger plant, averaging 24 grams in weight, whereas genotype A produces a plant that averages 20 grams. C. In this example, there are clear differences in the two environments. Both genotypes produce heavier plants in the wet environment. D. There are also differences in the weights of the two genotypes, but the relative performances of the genotypes depend on whether the plants are grown in a wet or dry environment. E. In this case, the influences on phenotype cannot be neatly allocated into genetic and environmental components because the expression of the genotype depends on the environment in which the plant grows. F. The phenotypic variance must therefore include a component that accounts for the way in which genetic and environmental factors interact. 8. In summary, the total phenotypic variance can be apportioned into 3 components: IPP equals BG plus BD plus BGE. White circle if H2 is close to 0. Variation in the natural population is almost solely responsible for phenotypic variance. Narrow sense heritability white circle is calculated by dividing the genetic variation resulting from the additive genes by the sum of the phenotypic variance. White circle genetic variance BG is referred to as additive variance BA. Black small square BG equals BA plus BD plus BI. Black small square BI is interactive variance. BD is dominance variance. Black small square here BI is often negligible. Black small square BG is partitioned into BA and BD. White circle narrow sense heritability says nothing the absolute value of a characteristic or about the underlying genotypes that produce these differences. Types of heritability. Broad sense heritability white circle in quantitative genetics. The proportion of the total phenotypic variance are differences due to genetic factors. This proportion is called broad sense heritability. It is symbolized as H2. White circle phenotypic variance is due to the sum of 3 components, namely environmental variance BD, genetic variance BG, and variance resulting from the interaction of genetics and environments BGE. White circle therefore, phenotypic variance black small square BP equals BD plus BG plus BGE, where versus GE is often negligible. White circle broad sense heritability. Black small square. BG genotypic variance. H2. BP phenotypic variance. Equals. White circle if H2 equals 1.0. Environmental factors have had little impact on phenotypic variance. Mutation 9. For nucleotide pair per generation or the number per gene per generation. The mutation frequency is the number of occurrences of a particular kind of mutation, expressed as the proportion of cells or individuals in a population, such as the no. Of mutations per 100,000 organisms or the number per 1 million gametes. Some important terms. Lethal mutation. The mutation of gene which brings death to an organism. For e.g., curly mutation becomes lethal in homozygous conditions. Balanced lethal mutation. When one or more lethal genes remain in one organism, the stock may be kept viable, keeping the lethal alleles in a heterozygous condition. For e.g., curly lobe and plum alleles can be maintained in heterozygous form. Conditional lethal mutation. This is a condition when one of the two sexes becomes affected due to lethality. For e.g., not just a mutant allele which is lethal for males in homozygous conditions, but a heterozygous female can survive. Dominant mutation. The mutation which produces an allele having dominant effect on the normal allele. For e.g., the mutant allele for bar I phenotype B plus B plus B plus B is dominant over normal IBB allele. Genetic screen. A mutagenesis procedure in which essentially mutagenized progenies are recovered and individually evaluated for the mutant phenotype. The desired phenotypes are marked in some way to enable its direction. Genetic selection. Some mutagenesis protocols are cleverly designed so that only the desired phenotype survives or replicates. Such protocols are termed genetic selection. Introduction. Mutation is the process by which a sequence of base pairs in a DNA molecule is altered. A mutation may result in a change in either a DNA base pair or a chromosome. A cell with a mutation is called a mutant cell. If a mutation happens to occur in a somatic cell in multicellular organisms, it is a somatic mutation. In this case, the mutant characteristic affects only the individual in which the mutation occurs and is not passed on to the succeeding generations. In contrast, a mutation in the germline of sexually reproducing organisms is called a germline mutation. This type of mutation may be transmitted by the gametes to the next generation, producing an individual with a mutation in both its somatic and germline cells. Any detectable, inheritable qualitative or quantitative change in the genetic material of an organism not caused by genetic recombination is known as mutation. Mutation. Changes in the genetic material, including the method or process by which a change has occurred. Mutation is the process by which a change in the chromosome or change in DNA base pair occurs. Mutations at the level of the chromosome are called chromosomal mutations. Mutation in the sequences of genes at the level of base pair are called gene mutations or point mutations. Mutant. An organism exhibiting a novel phenotype as a result of a mutation is referred to as mutant. Mutagen. Any physical or chemical agent that significantly increases the frequency of mutational events above the spontaneous mutation rate is called mutagen. Two terms are used to give a quantitative measure of the occurrence of mutations. The mutation rate is the probability of a particular kind of mutation as a function of time, such as the no. Of mutations leaky mutation. A mutation that confers a mutant phenotype but still retains a low but detectable level of wild type function. Genetic dissection. The use of recombination and mutation to piece together the various components of a given biological function. Paramutation. An epigenetic phenomenon in plants in which the genetic activity of a normal allele is heritably reduced by virtue of that allele being heterozygous with a special paramutagenic allele. Phenocopy. An environmentally induced phenotype that resembles the phenotype produced by mutation. Different types of mutations. Nature of mutation type. 1. According to the size and quality. A chromosomal mutation I. Variation in number. P. Variation in structure. B. Point mutation. 2. According to direction of forward B. Reverse. 3. According to magnitude and phenotype. A. Dominant. B. Recessive. C. Lethal. 4. According to origin of spontaneous. B. Induced. 5. According to cell type of somatic B. Gametic. Table 1. A gene or chromosome that differs from a wild type, an arbitrary benchmark for what constitutes an organism's normal, is the result of a mutation. Changes to the gene or the chromosome itself may be the cause of the mutation. Therefore, a general definition of mutation might be gene mutation, point mutation, where a gene's allele changes. Chromosome mutation is a condition in which individual chromosomes, sets of chromosomes, or complete chromosomes alter. Somatic mutations. Mutations that exist in the body's somatic tissues. Progeny do not inherit mutations. Whether a mutation is dominant or recessive determines how much an impactable organisms can undergo artificial mutations by being exposed to abnormal environmental factors like radiation, particular physical conditions such as temperature and chemicals. Forward mutations. Forward mutations are those that occur in an organism and shift the phenotypic from the wild type to an aberrant one. Forward type mutations predominant. Back or reverse mutations. The error-correcting mechanisms frequently fix the forward mutations, transforming the aberrant phenotype into the wild type phenotype. DNA can be changed in several ways, including through spontaneous changes, errors in the replication process, or the action of radiation or particular chemicals. Chromosomal mutations, changes involving whole chromosomes or sections of them. Point mutation, a change of one or a few base pairs. A point mutation may change the phenotype of the organism if it occurs within the coding region of a gene or in the sequences regulating the gene. Gene mutations, mutations that affect the function of genes. A gene mutation can alter the phenotype by changing the function of a protein. Have on the phenotype, dominant mutations generally have a greater effect. Whether something happens early in development or later determines how much an impact it has on phenotype. Early rising mutations have a greater effect. Germinal mutations. Mutations seen in the body's embryonic tissues. Progeny may be exposed to mutations. Following the mutation, the first generation exhibits dominant mutations. Males with an X-linked mutation will exhibit a muted phenotype if an X-linked mutation is present in the female gamete. Recessive mutations can go unnoticed for many generations because they can only be detected through chance mating with someone who also carries the recessive allele. Spontaneous changes. The origin of spontaneous mutations, which appear spontaneously in nature, is unknown. They are also known as background mutations and have been observed in a variety of organisms, including Drosophila, mice, men, maize, red moles, bacteria, and viruses. Induced mutations. In addition to naturally occurring spontaneous mutations, biological. Figure 1. Point mutations fall into two general categories. A base pair substitution mutation, change from one base pair to another in DNA, and there are two general types. White circle transition mutation, a mutation from one purine pyrimidine base pair to the other purine pyrimidine base pair, such as A to GC, purine purine and pyrimidine pyrimidine. White circle transversion mutation, a mutation from a purine pyrimidine base pair to a pyrimidine purine base pair, such as GC to CG or AT to CG. Purine pyrimidine and pyrimidine purine. Base pair substitutions in protein coding genes also are defined according to their effects on amino acid sequences in proteins. Depending on how a base pair substitution is translated via the genetic code, the mutations can result in no change to the protein, an insignificant change, or a noticeable change. Figure 2. White circle missense mutation, a gene mutation in which a base pair change causes a change in an mRNA codon so that a different amino acid is inserted into the polypeptide. A phenotypic change may or may not result, depending on the amino acid change involved. White circle nonsense mutation, a gene mutation in which a base pair change alters an mRNA codon for an amino acid to a stop nonsense codon UAG, UAA, or UGA. A nonsense mutation causes premature termination of polypeptide chain synthesis, such shorter than normal polypeptide fragments, often non-functional or released from the ribosomes. Figure 3. A nonsense mutation and its effect on translation. Figure 4. Subset of missense mutations in which a new codon codes for a different amino acid that is chemically equivalent to the original or the amino acid is not functionally important and therefore does not affect the protein's function. White circle neutral mutation, a base pair change in a gene that changes the codon in the mRNA such that the resulting amino acid substitution produces no detectable change in the function of the protein translated from that message. A neutral mutation is a figure 5. White circle silent mutation, a silent mutation, also known as a synonymous mutation, is a mutation that changes the base pair in a gene, but the altered codon in the mRNA specifies the same amino acid in the protein. In this case, the protein has a wild type function. Silent mutations most often occur by changes such as this at the third wall position of the codon. This makes sense from the degeneracy patterns of the genetic code. Figure 6. White circle frame shift mutation. If one or more base pairs are added to or deleted from a protein coding gene, the reading frame of an mRNA can change downstream of the mutation. Addition or deletion of one base pair shifts the downstream reading frame by one base so that incorrect amino acids are added to the polypeptide chain after the mutation site. It usually results in a non-functional protein. It may generate new stop codons, resulting in a shortened polypeptide. They may result in longer than normal proteins because the normal stop codon is now in a different reading frame, or they may result in a significant alteration of the amino acid sequence of a polypeptide. Figure 7. Point mutations are divided into two classes based on how they affect the phenotype. 1. A forward mutation changes a wild type gene to a muted gene. 2. A reverse mutation, reversion or back mutation changes a muted gene at the same site so that it functions in a complete wild type or nearly wild type way. A reversion of a nonsense mutation occurs when a base pair change results in a change of the mRNA nonsense codon to a codon for an amino acid. If this reversion is back to the wild type amino acid, the mutation is a true reversion. B. If the reversion is to some other amino acid, the mutation is a partial reversion, and complete or partial function may be restored, depending on the change. Reversion of missense mutations occurs in the same way. C. The effects of a mutation may be diminished or abolished by suppressor mutation, i.e., a mutation at a different site from that of the original mutation. D. A suppressor mutation masks or compensates for the effects of the initial mutation, but it does not reverse the original mutation. Suppressor mutations may occur within the same gene where the original mutations occurred but at a different site, intergenic suppressors, or they may occur in a different gene, intergenic suppressors. E. Both intragenic and intergenic suppressors operate to decrease or eliminate the deleterious effects of the original mutation. F. Intergenic suppressors act by altering a different nucleotide in the same codon where the original mutation occurred or by altering a nucleotide in a different codon. G. Intergenic suppression is the result of a second mutation in another gene. Genes that cause the suppression of mutations in other genes are called suppressor genes. For example, in the case of nonsense suppressors, particular turn genes mutate so that their anticodons recognize a change-terminating codon and put an amino acid into the chain. H. Thus, instead of polypeptide chain synthesis being stopped prematurely because of a nonsense mutation, the altered suppressor turn inserts an amino acid at that position, and full or partial function of the polypeptide is restored. This suppression process is not very efficient, but sufficient functional polypeptides are produced to reverse or partially reverse the phenotype. There are three classes of nonsense suppressors, one for each of the stop codons UAG, UAA, and UGA. For example, if a gene for a tyrosine turnout, which has the anticodon 3' August 5' is mutated so that the turnout has the anticodon 3' AUC 5', the mutated suppressor turnout, which still carries tyrosine, reads the nonsense codon 5' UAG3. Mutation of a ternate gene. A ternate gene has mutated so that the anticodon of the ternate is changed from 3' August 5, which can now read a UAG codon, inserting tyrosine in the polypeptide chain of that codon. H. So, instead of the chain being terminated, tyrosine is inserted at that point in the polypeptide. Mechanism of action of an intergenic nonsense suppressor mutation that results from the figure 8.